Okay, so welcome to this next video on the GQ pathway and calcium signals. So we have looked at calcium blips, we have looked at calcium puffs, these two types of calcium signal that can be given rise to by activating the GQ pathway within cells. In the case of a calcium blip, you need a tiny and very local activation of the GQ pathway. In the case of the calcium puff, again, you need a local but larger rise in the activation of the GQ pathway. Now what we're going to look at is the calcium wave, which is where you need a global, a whole cell activation of the GQ pathway. So calcium wave. Okay, so basically what happens is, if we draw a picture of a whole cell, because this time it really is a whole cell calcium signal that's going to occur. Okay, so if we draw a picture of the whole cell then. So, if this is the endoplasmic reticulum here, that's sort of spreading out through the entire cell, then what gives rise to a calcium wave is if you have activation of the GQ pathway in the whole cell, basically. So you need to have your ligand binding to your GPCRs and activating the GQ pathway all over the cell, basically. So what's the result of this activation? Well, you get IP3 being produced all over the place. So IP3 goes up in the cytoplasm of the cell all over the place. So you get a global or a whole cell rise in IP3. Now, this means that every single IP3 receptor in this endoplasmic reticulum is likely to bind IP3 to it. So all four subunits of the IP3 receptor are going to bind their IP3, and what that is going to do is it's going to mean that all of these IP3 receptors have available stimulatory calcium binding sites, basically. So, all that it takes now is for one IP3 receptor to just happen to bind 4 calciums in its 4 stimulatory calcium binding sites, and then one of them will open. So let's say this one just happens to achieve that, basically. Then what will happen is it will open, it will release calcium from the endoplasmic reticulum, calcium will go up in its local vicinity, and what will happen is some of this calcium will spill over onto the next IP3 receptor, basically. Now, this IP3 receptor, again, has 4 IP3 molecules bound to it because the IP3 has gone up globally and therefore has 4 stimulatory calcium binding sites available. So calcium is going to bind, you're going to get 4 calciums binding, 1 in each of the stimulatory calcium binding sites. That will cause the IP3 receptor to open, and you'll get calcium going from the endoplasmic reticulum into the cytoplasm. So you're going to get this one releasing calcium. So you'll go from having calcium being released here to causing calcium being released here, and then what will happen is calcium from here will spill over onto the next one. So you'll get this propagation in this release of calcium. Okay, this is what is known as a calcium wave, this wave of activation of the release of calcium from the intracellular store. Now, what will happen is, as calcium builds up over here, what will happen is that the calcium will start inhibiting that IP3 receptor, and therefore the IP3 receptors over here will start to close. So, if we were to measure calcium level in this portion of the cytoplasm here, what we would find, so let's say this is position 1, what we would find is if we plotted a graph of calcium concentration versus time, so let's say this is time, this is calcium concentration here, then what we would find is something that looked like this. Calcium concentration would initially go up when calcium was being released by the IP3 receptors in the vicinity of here, in the vicinity of point 1, and then it will gradually go down as these IP3 receptors are all inactivated. Then, if we were to look at calcium concentration in position 2 and plot it on the same graph, so this is position 1, what we would see is that as calcium goes up here, the calcium was spilling over, activating the stimulatory binding sites on this neighboring IP3 receptor and causing those to open. So calcium would be going up slightly later, basically, at position 2, like so. And again, it will be falling down as calcium builds up around these IP3 receptors and then starts to inhibit them, basically. Okay, and then if we did it finally for position 3 over here, then as calcium spills over from position 2 onto position 3, then it will bind to these stimulatory calcium binding sites, which are available because you've got 4 IP3 molecules bound to you, and that will cause the release of calcium from the intracellular stores here, and you'll get calcium rise, and it might have a position like that. So basically you get this spreading wave of calcium release. Okay, now there's more. Basically, in the case of calcium waves, this doesn't just happen once, because what if I keep the ligand bound to hit this GPCR? What if I just keep IP3 levels high for an extended period of time? Do I just get one calcium wave and that's it? No, absolutely not. What happens is that the calcium wave, after it's completed, after it's spread along through the cell, what happens is you get it starting over again. And this is the bit that is not understood, basically. We understand how one calcium wave happens, but what is not understood is how the second one happens. I mean, it's understandable that as calcium goes down, what's going to happen is that the calcium's going to go from having an inhibitory effect at very high concentrations to, as the calcium goes down, because the Surca pump is pumping it back into the endoplasmic reticulum, the calcium will go. It's understandable that the calcium will go back to having an activatory effect, and that might propagate it all off again. But what's not understood is that the frequency of these calcium waves is determined by your IP3 concentration. So the higher your IP3 concentration, the smaller the time interval between calcium waves, basically. So remember, frequency of a wave just means how many waves you get per second equals number per second. So if you've got a higher frequency, that means you get more waves per second, i.e. that the time between consecutive waves is smaller. So the higher your IP3 concentration, which is obviously indicative of how much stimulation you're getting from the ligand, i.e. how much stimulation of the GQ pathway you're getting. So the more ligand, the more activation of the GQ pathway, the higher your IP3 concentration. Anyway, the higher IP3 concentration, the faster the waves follow each other, basically. The smaller is the time between consecutive waves. So let me show you this with a picture. So let's say we plot calcium waves. So we'll plot the concentration of calcium at position 1. So in the first calcium wave, it goes up like so. And then maybe what will happen is the second calcium wave, there'll be some gap between the first calcium wave and the second calcium wave, and then there'll be a second calcium wave. Okay? But if IP3 concentration is higher, what will happen is you'll get the first calcium wave, and then the gap between the first and the second will be smaller, basically. So you'll get more frequent calcium waves if the IP3 concentration is higher. And this is how stimulus intensity, how concentration of the ligand you're being exposed to is encoded in the cell. It's not encoded in how high these calcium waves get, i.e. the actual amplitude of the wave, or how high calcium gets in the vicinity of these IP3 receptors. That is a set quantity. The IP3 receptors will allow their concentration of calcium nearby them to get to a certain level before that calcium starts having a negative feedback. So you can't raise the amplitude of this calcium spike, basically, within the wave. Instead, the intensity of the stimulus is encoded by how frequent the calcium waves are, rather than by amplitude. And that's a very good way of doing things, because if you encoded it through the amplitude of a calcium spike, then you could get dangerously high calcium levels within this cell. Remember, calcium is very toxic to cells. That's why they keep it so low within their cytoplasm. So encoding with frequency rather than amplitude of the calcium spike in the calcium wave is a good idea, basically. Also, it means that the signal is basically digital rather than analog. Very much so like action potentials, where you can either fire an action potential or you don't fire an action potential, and then the intensity of the sensory stimulus is encoded by the frequency of the action potentials fired along a neuron, rather than by how big the action potential is. Indeed, the action potential is always the same size. In the same way, a calcium wave is always the same size, and the stimulus intensity has to be encoded by how frequently these calcium waves fire, basically. Okay, but the mechanism by which IP3 concentration determines the time period between consecutive calcium waves is not understood, basically. But that's what a calcium wave is. Those are the three types of calcium signals that you can get from stimulation of the GQ pathway.